The central dogma of biology is DNA goes to mRNA, which goes to proteins. An organism's genome is encoded as a DNA sequence, and the proteins that DNA eventually make is the key to all life and the entire cell's function. Our DNA sequences are a repeating sequence of 4 letters, A, T, C, and G, and these letters are read in batches of 3 called codons to make amino acids. Stringing these amino acids together is what creates a protein. Yet simple math shows us that there are 64 possible codons and there are only 22 amino acids. This is solved by having synonymous codons, which means multiple codons which code for the same amino acid. However, recent research has shown us that synonymous codons are not redundant. Their physical structures and other relationships affect gene expression and affect protein folding. In fact, a study by Welsh et al. Showed an even 100-fold difference could be created just by changing which synonymous codons are used. This is incredibly important for the field of synthetic biology, where tools which design synthetic plasmids, or tools used to translate plasmids from one organism to another, can be improved significantly through accurate codon selection. However, today, many of the codon optimization programs simply look at which codons occur at the highest frequency in the host genome. They believe that by replicating that frequency, it would be similar to the host genome and would therefore be more efficient. However, research has challenged that assumption. A Morrow et al. Study showed rare codons are actually incredibly important for the function of the protein. This is because these rare codons often slow down translation at key steps, allowing complex secondary structures like beta barrels to form. And by removing these rare codons, which is what many of the industry tools do, we may actually be negatively impacting efficacy. And these proteins that we design may be different than what the cell usually designs, causing toxicity. In this project, I designed a new codon optimization tool, with the hypothesis that learning sequential information, the sequence of the amino acids, might yield important information about which synonymous codons to select, and it may allow us to better mimic the host genome. My solution to encoding sequential information was to utilize recurrent neural networks, or RNN. I developed a tool which could encode an amino acid sequence into a vector and then pass that through a series of layers in a RNN model, and finally output a DNA sequence with an optimized codon selection. I trained the tool using a newly compiled dataset of over 37,000 proteins from the NCBI bacteria database for E. coli. This database was incredibly generalized over all species of E. coli. The database was also cleaned by removing redundant proteins to ensure that no biases were created for high-frequency proteins. The final ICOR architecture involved a bi-LSTM layer, meaning a bidirectional LSTM, followed by a fully connected layer, followed by a ReLU layer, a softmax activation layer, and finally a classification layer. The end result is a tool which could import an amino acid sequence or a DNA sequence by a user and output a sequence which better matches the codon biases of the host organism, in this case E. coli bacteria. This tool has applications as a standalone tool, so that way users can improve their synthetic plasmids before those synthetic plasmids are produced and synthesized. Or the ICOR tool could be used in an existing product such as Eugene or other tools that are used for the development of synthetic plasmids to continuously check the developed plasmids and improve them before they are synthesized. Let's walk through a demo of ICOR in action. Here's the ICOR EXE. It is essentially a program which users can install locally to their computer. All the user has to do is import a FASTA file with their amino acid sequence that they would like to be optimized. They can also input it as a simple text file. Then they select which network to use. Right now, the current version of ICORNet is posted on the GitHub repository. However, further improvements in ICORNet can simply be posted online and users can download them as these improvements become available, thus making an incredibly lightweight and fast way for users to be able to perform codon optimization. Finally, ICORNet will predict the optimized codons and output them, both in a string format and as a text file for users to use in other applications. The whole process is incredibly fast, beating or at least matching many of the industry standard tools. Overall, the ICOR tool shows great promise, as it showed an accuracy of 54%. This accuracy is a 2.5-fold improvement over randomly selecting a synonymous codon. The development of this tool shows great promise for the use of sequential information to improve the synonymous codon selection. The ICOR tool essentially had to learn from scratch. It wasn't told which synonymous codons belong to which amino acid, and it simply had to classify for 64 classes. That means through this training, it was able to improve its accuracy from 1% to about 54% just by learning sequential information. Further, the ICOR tool is likely underfitting based on processing power limitations and validation data. This means that improving the complexity of the model by adding more processing power could allow for higher accuracy and even more optimized codon selection. ICOR also compares favorably to industry standard tools, especially considering the incredibly generalized data set it had learned from. ICOR also has the benefit of having same input, same output functionality, meaning that if a user inputs the same sequence, they will always get the same output, improving standardization of parts for synthetic biology. Other tools, such as the IDT standard, give a different output even with the same input, as they randomly select high-frequency codons. Let's walk through what the training and testing process looked like for ICOR in the MATLAB environment. First, the entire data set of training data was imported as a FASTA file. This was translated through a custom script which created the amino acid sequence as well as the nucleotide sequence in a structured format. Then it was passed through another custom script which created the training data format by encoding the amino acid letter sequences into vectors. The data was then split 80% for training, 20% for testing, and was then passed through the ICOR network architecture. Finally, the accuracy was analyzed through a custom getAccuracy script. The ICOR tool required many iterations and optimizations. The art of optimizing hyperparameters and networks is not really a science yet, but an art. When comparing with Presync Codon, another industry tool, on the EGFP reporter sequence, we showed a 78.8% similarity in the nucleotide sequence of ICOR compared to Presync Codon. In the Presync Codon research, they had shown that their tool showed a 2 to 3-fold improvement in the expression of EGFP by optimizing for low-frequency codons in addition to high-frequency codons. Due to ICOR's similarity to the Presync Codon, we may expect similar improvements or perhaps even better ones. In the future, I would have liked to synthesize the ICOR suggested optimized codon sequences and done an experiment to measure the efficacy by using EGFP and other reporter sequences.